Welcome to the Garden Talk podcast, where we interview growers from all over the world, both beginners and experts, seeking to learn more about what they know about gardening and how they do things in their garden. What's up, everybody? For you that don't know me, my name is Chris, aka Mr. Grow It, and you're tuned into the Garden Talk podcast. This is episode number 129. In this episode, I interview Dr. Tassa Saldi. She is the Chief Science Officer of TUMI Genomics and is an expert in molecular biology, with a focus on RNA biology. Within this episode, she talked in detail about a pathogen that has infected a large percent of both commercial facilities and home gardeners around the world. Everything from what the pathogen is, how it's transmitted, its symptoms, and so much more. If you want to see short clips of these podcast episodes, search Garden Talk Clips on YouTube. I also have a gardening channel where I show the plants that I grow. I'll have both those channels linked down in the YouTube description section below. One of my goals for this podcast is to bring free information about gardening to the general public. That being said, I'd like to thank the sponsors of today's episode who help make that goal possible. Thanks to AC Infinity for sponsoring this episode. Check out their Controller 69, which can help automate your environment's conditions. It'll automatically turn on and off equipment to help control the temperature, humidity, VPD, light schedule, and even the oscillating fans. Click the link in the description section below so you can learn more about the Controller 69. And the discount code MrGrowIt15 works on both Amazon and their website, acinfinity.com. Stash Blend. Stash Blend consists of several ingredients in one, so you no longer have to buy individual bottles. It includes corn steep liquor, seaweed extract, humic acid, beneficial bacteria, silica, and mycorrhizal fungi. It can be used in addition to your synthetic base nutrients or be used in living soil systems. Simply mix it into water and water your plants with it. You can get it at stashblend.com, link in the description section below, and use the discount code THESTASH. And we are back. Welcome to the Garden Talk podcast. Today I am joined with Dr. Tassa Saldi. How are you doing today?
I'm good. How are you?
Doing pretty good. Thanks for coming on to the podcast today. Today we're going to get into an issue that's wiping out commercial facilities around the world. 73% of commercial facilities in the U.S. have been infected. And just in California alone, 90% of cultivation facilities research tested positive for this viroid. What we're talking about today is hop latent viroid. And it's actually pretty scary, for a number of reasons, which we're going to get into in this episode. But first, let's do an introduction. Can you tell us a little bit about yourself and how you got into gardening?
Yes, absolutely. So my name is Tassa. Like you said, I am the Chief Science Officer of TUMI Genomics. So my background is in molecular biology and specifically in a type of biology called RNA biology. And what my background looks at is how the shape or the structure of certain molecules can impact cellular metabolism. And so this is pretty on point for hop latent viroid because this particular pathogen enacts its pathology based on its three-dimensional shape or its secondary structure. And so that's what I did my graduate and postdoc work on. So we founded our company, TUMI Genomics, a little over 4 years ago. And we moved into helping cultivators identify pathogens and especially this particular pathogen maybe about 3 years ago. And basically we had somebody who was running a nursery contact us and ask us if we could help them identify this particular pathogen. And we were all molecular biologists actually working on, we started in cancer and then we moved into COVID. But we agreed, we could definitely help identify this as well. And once we realized the breadth of this issue and how critically the cultivation industry needed to have good diagnostics, more research to understand this particular pathogen. We kind of found our home as a company. And so that's what we've been doing for the last 3 years is, research collaborations with academia, with industry, as well as developing, reliable diagnostics and even some on-site testing to help cultivators identify this thing and keep their operations safe.
Awesome. So what exactly is hop latent viroid? Break it down for us and mention how does it differ from other pathogens?
Yes, absolutely. So, hop latent viroid and viroids in general are one of the strangest and I would say most interesting organisms, if you can even call them organisms, out there. So, most organisms contain, well, almost all of them except for viroids, contain an inside part which has their information. So, that's like your DNA, or in viroids' case, it's RNA, which is a molecule very similar to DNA. But on the outside of that information is like a coating that keeps the genetic information or the DNA RNA safe from the outside environment. And that outside coating is generally made of proteins or fats or both. And that's the same if we're talking about funguses, bacteria, or even viruses. They all have a coating like that. So viroids are incredibly unique in the sense that they basically say, I don't need any of that protection stuff. And they're just literally free floating information. So when you think about viroids, it's much more helpful to think of them like an inert molecule or contamination at a crime scene instead of like a living organism. And they're incredibly, incredibly small. And this makes them very, very contagious and very easy to pass from plant to plant. They are very specific to land plants. So for the most part, except for a few kind of outlying publications, viroids are exclusive to plants. So they can't hurt people if you were to consume a viroid or, smoke something like a tobacco cigarette or something like that with viroid in it. But they can be incredibly damaging to plant development and especially plant flowers.
Got it. Now, people are hearing the word viroid, and I'm sure there's quite a few people out there like, viroid, you mean virus? So totally different. I know you kind of explained it. Is there anything else to be said in regards to the difference between a virus and a viroid?
Yes, absolutely. So because viroids are so incredibly simple, they don't actually have the ability to do any type of metabolism or even information to code for a metabolism that allows them to replicate. So viruses have a unique biology to them, which is why we can make things like antivirals, for example, because they're targeting something that is different in viruses than in the organisms that the viruses infect. But viroids are so incredibly simple that they don't have that unique biology. So everything that they do is hijacking the biology of the plant, which makes them incredibly difficult to target because it's very difficult for whatever we're targeting to differentiate between the viroid and the plant's normal biology. And so that's why, as opposed to a lot of other things, there aren't really any effective treatments or anything that would specifically target the viroid, at least yet. So they're very difficult to treat because of that.
Wow. And taking a step back and looking at the big picture, what's the economic impact from this viroid outbreak?
Yes, yeah. So it's huge. There's an estimate that for cultivation facilities across the board, there's more than a $4 billion loss annually. And that's, those estimates are just mostly what's going on in the United States. We've found at TUMI Genomics that this particular viroid is present on pretty much every continent. We've tested plant material from Canada, Africa, Asia, Europe, and it's pretty much everywhere. And like you mentioned at the beginning, it's in 70 to 80% of cultivation facilities. And you can see up to a 30% loss with plants when they are infected with this, which accounts to a huge amount of money and a pretty prevalent risk for every single cultivator out there. So it's truly a pandemic at this point in the industry.
That's incredible. Mostly on the home grow side of things. And I think a lot of my audience is, but we do have folks that are in commercial facilities, for example. But
Yep.
I can only imagine what the impact is on the home grow side of things.
Yes.
There is a lot of no testing happening, particularly I know that we're going to get into how it can be transmitted. I know seeds is one of them, but yeah, it's got to be widespread amongst so many folks, and they probably have it and they don't even know that they have it, right?
Yes.
Symptoms. Let's get into that next. plants that are infected with hop latent viroid, what are the symptoms that you can see on the plant?
Yes, absolutely. So hop latent viroid has, we call it, what it's called is pleiotropic, which means that there are a ton of different symptoms. And it basically affects every system of the plant. So different things that you can see. It affects the growth of the plant. So you can see the stunting of the plant. It also can affect the shape of how it grows. So as opposed to like a normal 45-degree angle of the branches coming off that main stem, when plants are infected with hop latent viroid, the stems tend to come out more horizontally. You can see leaf chlorosis, misshapen leaves. Sometimes the leaves will overlap. Sometimes they'll look too shiny, or depending on, the specific cultivar you're looking at, they can also kind of turn a little yellow or curled up. Rooting is another major issue for plants that have hop latent viroid. They tend to root very slowly. And the root ball itself is reduced in density. So if if you plant a clone and it's trying to root and it just taking forever, that's a really good sign that that particular plant may be infected with hop latent viroid. And then, of course, it also affects the development of flowers on plants. And so the flowers tend to be underdeveloped, reduced in mass, reduced in chemicals and things like that. And so it kind of takes down every system of the plant. And it renders it, whatever, especially if you're looking for medicinal properties from that particular plant, those different chemicals are not being produced. And so the plant is not nearly as useful from that standpoint.
Wow. So it hits the plant all over. And yeah,
Yes.
some of those symptoms that you mentioned can be done by other things, right? So you mentioned like chlorosis, for example. There's a number of problems that happen where you get yellowing leaves. Hop latent viroid is one of them. Leaf overlapping was an interesting one. I actually had a plant that had leaves overlapping, and I was like, oh no, hop latent viroid, because I had learned that in the past. And so I actually got that plant tested, and it actually came up negative.
Mm-hmm.
So I was a little bit surprised by that because I hadn't seen that in any other cultivars that I had grown. But yeah, the overlapping leaves was a really interesting one. And then, root development slower, smaller root ball, underdeveloped flowers, stunning of growth. There's just so many different things that this viroid does to the plant.
Yes, yeah. But now I should mention that one of the trickiest and most insidious things about this particular pathogen is that while all of those symptoms are possible, it's possible that either only some or none will manifest in the plant. And so even if you're looking at a plant and you're like, oh, this plant looks great, it looks super healthy, everything is fine, it's still possible that it's infected. It's definitely possible that it will appear asymptomatic during the early stages of growth, and you're not going to notice any changes until that plant grows up into maturity and goes into flower. And then often you'll see those symptoms then. And so it's important to understand what the symptoms could be, but it's also important to realize that you may have an infected plant and it looks pretty good. And so the only way to be sure is to get that test, like you said.
So how does hop latent viroid spread, and what measures can gardeners take to prevent its transmission?
Yes. Yeah. So hop latent viroid, like most viroids, is spread primarily by mechanical means. So that what this means for the most part is it's spread most efficiently by us. So if you're trimming your plants, for example, and you take those scissors, you trim a plant, and then you move to the next plant, and you don't clean those scissors first, that the sap from the potentially infected plant can infect the non-infected plant. And so that's the most common way that hop latent viroid is spread. Other potential ways that it's spread, it can be spread through the water. So hop latent viroid can slough off of root cells, or the root cells slough off and contain hop latent viroid. There's a lot of hop latent viroid in infected plants in the root tissue. And so if plants are sharing water, like if they're in a clonal tray, or you're doing some sort of flood and drain or some system where plants are sharing water, you can transmit it like that. It can also be transmitted through seeds. So there are several studies out now, and we've also conducted a study showing that seed transmission of this in medicinal plants is somewhere between 20% to 50%. It can also be transmitted in the pollen to a smaller extent. Our numbers put it around 5% of the time. If a male is infected, it can cause the seeds to be infected as well. And then the potential way is bugs. So this is research that's just coming out of CSU here in Colorado in Fort Collins, showing that there are bugs. I think their research was done on aphids, if I'm correct, that are capable of transmitting hop latent viroid from plant to plant. So pretty much the seeds, water, and anything that moves plant sap from one plant to another is a potential way that this thing can spread through a facility or between plants in a home situation.
So it's extremely easy to spread.
Yeah, it is very contagious, and it's very insidious because you can spread it and not notice for several generations. Like if you're cloning a plant, sometimes you're not going to see any symptoms until the second or third cloning, and then all of a sudden everything goes to hell and you don't understand. And that happened to us. We have plants as well, and before we knew about this, I think like everybody else out there, all of a sudden, the plants weren't flowering correctly, they didn't look right, and we had no idea what was going on. So it happens all over, from huge cultivation facilities down to, someone who's growing at home.
Wow. And that's where testing can be so important. And we're going to get into testing here in a little bit. But first, I do want to ask you, how long can viroids survive for?
Yes. Yeah. So even though they don't have that outside protective coat that I was discussing, they're actually incredibly robust. The shape of viroids it's a circle, so it's like closed at the end, and it's kind of like Velcro. So you can think of it like a Velcro circle that's smushed onto itself. So it's more like a rod shape than some sort of free floating like a noodle or something. And that shape affords a lot of stability. So studies right now show they only went out to 24 hours, but they're definitely stable for 24 hours or more on most common surfaces. So that would be wood, plastic, metal, cloth, whatever. They can survive for up to 7 weeks in water. So if you have water that's been sitting around and you keep putting plants in it, that's a potential spread there. And they can last for 3 months or more in dried up plant material. So it's really important that if plant material falls on the ground or you get sap on something, that you're cleaning that up right away. And then in seeds, we've tested seeds at this point that were more than 2 years old. And it does tend to decrease in frequency of a seedling that's infected over time, but you can still transmit it in the seed for a long time, years. So it's very stable.
Incredible. Yeah, I actually listened to one of your talks, actually a couple of your talks at Utah State University. I attended there for a cultivation certificate that I have. And I took notes on your talks. And you nailed it on there. A couple other things you mentioned there was greater than 90 days in fermented compost. And then you also mentioned several months in soil or growing medium.
Yes. Yeah.
Wow.
Yeah. Yeah. And it's part of the hardest part about soil is that hop latent viroid is incredibly high concentration in the roots. And so when you're removing plants from soil, it's really difficult to not leave some root material behind. And so that's one of the hardest things about growing in soil is that it's difficult to remove those infections once they've established in there.
Now, are there any plant species or cultivars that are more susceptible to infection?
Yes. So hop latent viroid, so in the wild, so in nature, it has a pretty narrow host range compared to most other viroids. It's infectious, obviously, in hops. That's where the name came from. It can also infect stinging nettle, and it can infect industrial hemp. So in lab settings, they've shown that they can also infect things like pea plants and tomato plants. There is yet to be any literature showing that that's happening out in nature, but it is a possibility. We also just completed a study looking at the susceptibility of different cultivars to hop latent viroid. And what we found was it was pretty interesting. There are some cultivars that are incredibly sensitive. So they're very easy to infect. They have really pronounced symptoms. And there's a huge amount of yield loss when they're infected. There's also a battery of cultivars that are what we call resistant, which means they were difficult to infect. The viroid didn't rise to a very high concentration until like the very end of the plant's life cycle, or when it's like finishing flowering. And then there was a few outliers, which I honestly find slightly scary, that were tolerant. So these were plants where they had an incredibly high level of infection, super easy to infect, spread very quickly, but the plants didn't show any obvious symptoms and there was no yield loss. So it's great. It's wonderful to have those plants because then you don't notice any problems, but they also are basically a Trojan horse. So you might think everything's great because that plant doesn't have any symptoms, but then it spreads it to everything else. So it can go all over the map depending on cultivar.
Jeez. Now, in regards to testing, what are the current methods for diagnosing hop latent viroid infection in plants? And are there any advancements in the detection technology?
Yes, absolutely. So the first important point about hop latent viroid and viroids in general is because they are only that genetic information, the only type of test that is capable of detecting them is basically a molecular test. If people were used to thinking about COVID, there was the antigen test and the molecular test, which was the PCR. So for viroids, it has to be one of those molecular tests. And the two major types of tests that are out there right now, there's a few exceptions to this, but the two tests that you're most likely to run into are what's called a PCR test or an isothermal amplification or a LAMP test. So both of these tests, the way that they work is they find the viroid in a tube, and once it's identified, the test creates a ton of copies of the viroid. So if there's 2 copies in the tube, what the test will do is make 4 copies, then 8 copies, then 16, then 32, and on and on. And so at the end of that copying, you can have billions of copies, and then it's easy to detect. The difference, so they both do that, PCR and LAMP. The difference between those two is that PCR is a much more high complexity test. It requires very expensive equipment that can cycle in temperatures, and it generally requires specialized expertise to understand the results. LAMP tests, on the other hand, are much simpler. They all run at a single temperature, and so that type of test or isothermal amplification is really appropriate for like an on-site or an at-home type of test. Currently, PCR tends to be more sensitive, more accurate, more reliable, and LAMP tests can often throw false positives, false negatives, and a lot of them don't contain an internal control, which means that you can't tell the difference between a failed result and a negative result. So what we've been working on at TUMI Genomics is kind of addressing that particular issue, because what we've been working on is trying to give an option to people that want something at home or on site that can allow them to find the viroid in their plants, but with a test that has the type of accuracy and sensitivity that you would need to be confident. So one of the things that we just launched at TUMI Genomics is an on-site testing platform called TUMI Glow. So it's based on isothermal amplification, but what we've been able to do with that particular test is increase the sensitivity to the same range as the PCR, the accuracy to the same range as the PCR, and include an internal control. So if you can tell the difference between the test is negative and I forgot to put plant tissue in the tube or something like that. So Yeah, we're really excited about releasing that or having people look into that, because I think this can be a very powerful tool in fighting back against this pathogen.
That's awesome. Sounds convenient for the average home grower to get testing done. Now, like I mentioned earlier on the podcast, I actually got a test done on one of my plants, and I'd like to walk through the testing process with you if you don't mind.
Sure.
So, if I remember correctly, this was last year, I had to take leaf samples off of, I believe, 2 or 3 parts of the plant. I think it was the top and bottom, maybe middle. And then also, I had to take a sample of the root as well and then send in for testing. Can you kind of walk us through that? Is that just for PCR, or is that for the isothermal test, or what's the testing process? What would a home grower do to submit in a test?
Yes, absolutely. So to submit a test, at least with TUMI Genomics, so we have specialized collection tubes that we mail out to people. The reason we do this is because those tubes contain a buffer in there that stabilizes the sample, so we don't have to worry about stuff degrading. It doesn't have to be sent on ice, nothing like that. When people are collecting samples, so we used to actually request that people collect some samples from the foliage and samples from the root, but we've recently switched to just requesting root. The reason this is that we've done a pretty comprehensive study that was then confirmed by a bunch of different groups that the most reliable and accurate tissue to detect this particular pathogen is in the root. So in the foliage, if you're looking for hop latent viroid, you have somewhere between a 12 to 60% success rate, something like that. In the roots, at least in our studies and what it appears to be in the other studies, is that it's close to 100%. If a plant is infected, you're going to find it in the root first. It has the highest level, and it's the most reliable spot. So when people are testing it with the PCR, they'll take root tissue, put it in a tube, close it, shake it up, and that's all they have to do. They'll mail it back to us. We can run the PCR in the lab and then send them results. For our on-site test, it's exactly the same. We require root tissue for hop latent viroid specifically. You put it in the tube, close it, shake it up, and then you take some of that liquid out, put it in the reaction yourself, let it sit in a heat block for some time, and then look at the tubes. And if it's positive, the tube will glow red to yellow, somewhere in that range, either red, orange, or yellow, depending on the viroid load. If it's negative, it'll glow green, and if the test didn't work, it just won't glow at all. So for both situations, we focus on root tissue because that's your best chance of catching it if the plant is infected.
Okay, and then what's the cost for testing?
Yes, yeah. So right now for the PCR test, for the cost is $25 a test. We do have bulk pricing if people want to get a bunch of tests, but the base cost is $25. For the at-home test, those start at $18 but come down very quickly. If you buy multiple tests or buy in bulk. So it just kind of depends on which of those two options you like better, but those are the pricing for those.
Okay. Yeah, $25 isn't bad. The other company I went through was double the price. So I'm going to have to go through you for now on. So that's
Yes.
TUMIgenomics.com, and then you can click order tests. I'm looking at it right now. Do you do any tests for anything besides hop latent viroid?
Yes, we do. We test. We also test for fusarium, all the pathogenic forms of fusarium in medicinal plants, as well as the pathogenic forms of pythium in medicinal plants. And so those are root pathogens that quite honestly can be just as damaging, and in the case of fusarium, perhaps even more damaging than hop latent viroid. And if you're growing indoors, then those three are the most likely 99% of all of your pathogen issues. So we also have tests that you can order one test and it'll test for all three of those at once. Which, if you're bringing in a plant, whether you have a facility or you grow things at home, it's really important to test and make sure that it's not infected, because the amount of time you're going to spend, months and all this material and your personal time growing it, and then to have it, die of fusarium or not produce well because of hop latent, it's absolutely worth getting tested. We do also offer a whole battery of viruses, those that are known to infect medicinal plants. Those are much more common if you're growing outside. So if you have a big buck issue or you're growing, even in your backyard or something like that, then those are significantly more common because they're spread by insects. And then we can also test water, growing medium, and air. So if you're having an issue, like if you have a facility and you have some sort of fungal issues and you think that it could be in your water, we can help you with that as well, or in cocoa or growing medium or however you're growing it. So in lab, we have a whole bunch of different things that we can help you with.
Okay, awesome. Good to know. Now, talking about strategies for managing or controlling hop latent viroid, right? So it's infected a plant. What do you do from there?
Yes. Yeah. So unfortunately, in the case of hop latent viroid, there is no cure. There's nothing you can spray on the outside of the plant to make it go away or fix it or suppress it. If you have a plant that is highly infected, your two options are to either toss it or to submit it for a process called tissue culture, which basically means that they clean up the plant by putting it through a series of tissue culture steps. Tissue culture does work, not 100%. It doesn't work on every single cultivar, and it's a very difficult procedure. So I know that there are, at-home tissue culture kits and things like that. This is probably not one of those situations where you'd be able to do this at home. The best way to prevent it, or the best way to deal with it, is to prevent it altogether. And that just involves not being afraid of it per se, but being aware that it's out there and that there are certain steps that you need to take to keep it from entering into your facility or your home or spreading from plant to plant.
Honestly, this is one of the things that really prompted me to clean my trimmers every time between plants. And I was doing it a handful of times, and now and then you hear about it. You hear it's the best practice, going to prevent pathogens and virus from spreading and so on and so forth. But as a home grower, you just go plant to plant, trimming, doing your training, high stress training or defoliation or whatever, and using trimmers and not cleaning them from plant to plant. But now, knowing that hop latent viroid is present, cleaning the trimmers between every single plant is something that I've been doing religiously now.
Yes,
and
very important. Yeah.
Just to confirm what you should be using for a solution. Now, some people say, okay, isopropyl alcohol, that's going to be good enough. Other people say, no, isopropyl alcohol isn't going to kill off the viroid. You want to use a bleach cleaning solution instead and dilute it at a 1 to 10, 1 part bleach, 10 parts water. What's your recommendation for that?
Yes, absolutely. So when we're talking about viroids, isopropyl alcohol or any alcohol is probably the worst thing that you can do. So it's not effective at all in killing viroids. And it actually, alcohol is one of the things we use to stabilize free-floating genetic information in the lab. And so it actually increases the transmission of the viroid from plant to plant if you use alcohol. So when they did these studies in, I believe it was tomato plants with a viroid, and they showed that if you do nothing, absolutely nothing, there's a 75% chance, if you don't clean those scissors, of spreading it. If you dip the scissors in isopropyl and then cut the plant, it goes up to 95%. So just throw the isopropyl out, use it for something else, because it's not a good idea for viroids. And to be totally honest, it's not 100% effective on bacteria or fungal pathogens either. So you're exactly right. If you're trying to limit the spread, bleach is incredibly effective. And that's that 10% solution that you were talking about, which is 1 part bleach, 9 parts water. When you are going to get bleach, you want to grab that bleach bottle from the Super Walmart or whatever and make sure that it has 6% of sodium hypochlorite or higher. So that would be the active ingredient. And then when you get that bottle home, put a date on it. And at 6 months, you should toss it and get another one because the activity will go down over time. It's very effective at killing viroids. So all you need is 60 seconds. It's probably less, but we recommend 60 in case there's like goo and stuff on the scissors. that it just needs to sit in there, and then the Viroid is completely deactivated. Usually I recommend if you're trimming a bunch of plants, just have several pairs of scissors. You can just grab one, the others are in the bleach. When you're done with one plant, you put it in the back, then you grab the next one in the front. And so you always have a supply of scissors that are clean. It is kind of caustic on your cutting tools. So if you're concerned about them or they're very valuable to you, put them in there for 60 seconds and then move them into like a cup of water. Tap water is fine. That will keep them from rusting.
Very good info. I appreciate you sharing that with us.
Yes, absolutely.
Now, you mentioned earlier it can last up to 7 weeks in water. And I vision those folks that are doing deep water culture, for example, like a recirculating system. So hypothetically, right, if one of their plants has hop latent viroid, it's in the roots, it gets dispersed into the water, all the plants are kind of being infected there. Are there any best practices in regards to like changing out the reservoir, or is it pretty much like almost immediate and there's no kind of getting around it as far as infecting the plant via the water in a deep water culture system?
Yeah, so there's no way to stop the viroid from sloughing off the root tissue and then entering into the water. And like you said, it's very stable in the water. The amount that is going to come off those roots is going to be really dependent on how infected that plant is. So if it's a low level infection, then obviously your risk to the other plants is lower. If the plant that is sloughing off hop latent viroid is basically on fire with viroid, then a massive amount is going into that water, and your chances of infecting the other plants is much, much higher. Recirculating systems, deep water culture, that's very difficult. And honestly, if you're growing that way, the best way to protect yourself is to just make sure that any plant you put in that system, you test first, that it's negative for hop latent and honestly, Pythium and Fusarium as well, which also spread incredibly easily in water. Pythium is a water mold. It loves the water. So on those types of systems, prevention is critical. I mean, it's critical for everybody, but for those types of things, it's even more important that you test first.
Okay. So I got to ask, what are commercial facilities doing about this, right? So they have an affected plant. Are they just killing off that plant and all their plants and starting fresh? Are there some commercial facilities that are continuing to grow out the plant? Because you said, there is a reduction in yield, but they'll still be able to get to harvest and it doesn't harm the consumer. You had mentioned earlier, it's like there's no, when you're consuming it, when you're smoking or whatever, it's not being transmitted to a human, for example. So are commercial facilities, are they killing off plant population and trying to keep their facilities clean? Or are you finding some facilities are actually just like, oh, well, okay, let me segregate this plant to a different room. Let me continue to grow it out. And then I just won't keep any of the clones off of these plants.
Yeah, so that's a great question. And the answer is it's kind of all over the map. And part of this is because there aren't clear regulations for cultivation facilities on how to handle this kind of thing, or guidelines either. And so we work with some facilities that are incredibly diligent about testing. Everything gets tested, every mother, just very, very on top of it, all the way to facilities that don't appear to test at all, and just aren't concerned with it. If there's a cultivation facility where plants are identified that have hop latent viroid, we have helped these facilities deal with this issue without having to basically, blowtorch everything and start over. And so the best way to deal with this is to consolidate your genetic library so that you have 2 to 3 plants of each cultivar. And you segregate those plants into a different space in the facility, which we call like the Garden of Eden or your like nuclear stock. And those plants are tested religiously and protected from the rest of the facility. So what you can do is slowly move plants from your genetic library that are clean into this special Garden of Eden while you continue your operations downstream. And some of those plants may be infected. But you basically kind of let them fall off the end and go into flower, and you're slowly building up this genetic library. And eventually all the plant material will come from this Garden of Eden, and it will start clean and hopefully stay clean through the entire process. There are definitely facilities that they don't have a choice. They have to flower the sick plants because they have quotas to meet and it's a business and they have money to make. But a lot of times, if you getting like lower quality type stuff, it's probably infected, because it's incredibly common. So that's what, if it's the lower quality kinds of things, it's probably because it's infected with hop latent viroid.
Okay. And then on the home grow side of things, would there be any different advice? Like, what advice would you have for those gardeners who receive a positive test for hop latent viroid on one of their plants in their garden?
Yes. So from a home grower perspective, because tissue culture is so expensive and extensive, I would just get rid of it. It's not worth your time or your money to grow that plant. And we did do a little experiment ourselves where we just went out and bought a bunch of clones, and just to see what we would get from different spots, and pretty much every single one of them was infected with something. Now, granted, we were probably picking ones that didn't look terribly happy, but if I would definitely, when you bring in any type of new material, test it right away. Quarantine it. Don't plop it right next to your favorite plants or your favorite strains or cultivars. Test it first in a separate area. If it comes back positive, just go back to where you bought it and say, hey, plant is sick with something. Give me another one or my money back or whatever. But whatever you do, do not start growing that with the rest of your plants. Don't start sharing tools and water or whatever until you confirm that it is negative. And just to be incredibly safe, I would try to get 2 negative tests, 2 to 3 weeks apart, 3 to 4 weeks apart, somewhere in that range, just to make sure that they didn't have a low level infection in the beginning. It's just not worth growing it. If you calculate the amount of money it takes to grow up a plant and your time and the lights and the electricity and the nutrients, and then what you have to do if it fails, and doesn't produce any medicinal properties, then it's just not worth the money to not test.
Got it. And I should ask this earlier when we were talking about testing, but are you able to test seeds?
Yes, we can test seeds as well. And seeds are an interesting thing because the flower of the plant tends to have one of the highest levels of viroid. And so if you have a package of seeds, you don't have to test, all of them, because sometimes they're pretty expensive for one package. So one seed from that package is sufficient for us to determine whether or not they came from a mother or a father that had hop latent viroid. But I would definitely test your seeds as well, because that's another way that it can get into your garden.
Great to know. Is there anything else that we need to know about hop latent viroid?
I would say just that I think there's a lot of fear out there right now in the industry about this particular pathogen. And it's important to know that this isn't something that people need to be afraid of. This is just something that people need to educate themselves on, understand how it works, understand how it's spread, understand how to prevent it, and then just put those systems into practice. Make sure you're keeping everything sterile and clean. Don't let anything into your garden unless you've tested it. If you're growing a lot of plants, routine testing is probably a good idea. If you're just growing a few, in your house or your garden, then, at least test them before you start growing them to make sure that they're going to yield what you're looking for. Just remember, prevention is the smartest way to go about keeping this thing at bay. Stopping it from entering in the first place is a whole lot easier and a whole lot cheaper than fixing a problem after it's happened. So, there's a solution to it. There's no reason to be afraid of it. Just protect yourself, and it's manageable.
Some good advice. Now, there are many other pathogens, whether it be a virus, a viroid that's out there that can infect a wide scale of plants, right?
Yeah.
We won't get into them here, but can you kind of leave the audience here with a little bit of a cliffhanger? What are some other pathogens out there that people can look into and research? I know there's beet curly top virus, there's lettuce mosaic virus, there's tobacco mosaic virus. Do you have maybe a few more that you can list off that people can look into?
Yes, absolutely. So some of the other viruses that have published evidence to infect medicinal plants are, like you mentioned, beet curly-top virus, lettuce chlorosis virus, tobacco streak virus, alfalfa mosaic virus. Tobacco mosaic virus, a lot of people are afraid of this one. There's no published evidence that it infects medicinal plants naturally, and no published evidence that it can cause symptoms in plants, in medicinal plants. So I would say that those 4 that I mentioned are the biggest threat currently. They're all spread almost exclusively by insects. So this is something that's much more, like I said, on point if you are growing plants outside. Other potential pathogens, especially viroids that exist in hops, which have the potential to jump over into other plants, are apple fruit crinkle viroid, hop stunt viroid, and crackling bark viroid. So all 3 of those, while there's no evidence they're in medicinal plants right now, it is highly possible that those things could jump across species. So what's most important is that you identify a lab where you're getting things tested that is kind of monitoring that stuff and has the scientific chops to develop tests quickly if those things become a threat in the industry. So basically the important thing to remember here is no matter what you're facing, if you prevent entry, if you're careful with sterilization, if you're paying attention, you can protect yourself from both the known pathogens as well as anything that might be coming down the pike. It's just important to put those protections in place, and it'll be all right.
All right. Well, so much good info in this podcast episode today. Thank you so much for coming on here. This is actually a topic that we haven't gotten into on this podcast yet. So I'm really honored to have an expert on here that can break it down for us and give us all the details and what we need to know and how to test symptoms and all that stuff. So I appreciate your time today. Can you tell the listeners how they can find you and maybe talk about some things that you have upcoming in the future?
Yes, absolutely. So you can find us at tumigenomics.com. We are also on Instagram at tumigenomics, LinkedIn. You can go to our website. There's all sorts of ways to get a hold of us. You can send us an email. There's a phone number. There's even a live chat. If you just want to plop in there and ask a question about hop latent viroid, I'm always happy to just help out with questions. Even if you're not ready to test, you just want to understand the process better, please reach out and get in touch with us. Like I said, things that are coming out or just recently came out would be our on-site testing platform. So you can also reach out to us and ask us questions about that, like how to obtain it and understand it a little bit more. We are also working on developing, hopefully soon, what we call a next generation sequencing type diagnostic. So what this can do for you is, instead of you knowing what's wrong with your plant, you can say, this plant doesn't look right. I'm going to send in some tissue, and we can basically agnostically look at it and tell you what's there. So you don't have to know that it's hop latent viroid or know that it's lettuce chlorosis or whatever. We can kind of test for everything all at the same time. So that's something else that we're hopefully coming out with soon. So people can just look for, if they don't know what's wrong, they can look for whatever. So those are the things that hopefully will be out in a reasonable period of time.
Awesome. Well, I'll definitely have a link to you down in the YouTube description section below, so people will easily be able to navigate to you. Thanks so much once again for coming on to the podcast. I really appreciate it, and hope you enjoy the rest of your day.
Yes. Yeah. Thank you so much for the opportunity to speak with you, and you as well. Have a wonderful day.
Peace out, everyone. Catch you in the next episode.